Well, good morning, everyone. Good afternoon and good evening for those of you who are joining us from various points all around the world. My name is Brittany Choate, and I'm Director of Programs for Saliva-Direct Inc. It's my pleasure to welcome you to this session of Saliva-Direct's fourth annual conference, that function at the bottom of your screen. As I said, I'm very pleased to introduce our first speakers, which will be presenting as a pair. Dr. Aaron Palmon and Dr. Omer Deutsch are joining us from Salignostics over in Israel. So we really appreciate them taking the time to join us at a later time in their day. Their presentation is on the state of the art, the development of saliva-based HIV and Strep A tests. Aaron and Omer, over to you.
I would like first to thank the organizers for inviting us, Omer and myself, to present our work. The importance of all fluid-based diagnostic tools was floated several times, and for us, the 202 NIDCR call for salivary diagnostic technologies for early disease detection and to monitor individuals' risk of exposure to harmful biological and other agents was an opening for new research efforts. Our long-run aim was to set a new platform for an easy, low-cost, rapid, and at-home saliva-based diagnostics. And for that, we asked for the development of new platform that will be chairside, simple to use, rapid, procedure takes up to 15 minutes, similar sensitivity to regular tests, affordable, and allows digital home scanning for telemedicine. This need is pronounced by the paradox of home use diagnostics, which use blood, urine, or feces, all not comfortable for home use. Saliva suggests much better sample for that. Though saliva is optimal for that, it also presents several challenges as low biomarker concentrations, masking characteristics, reducing detection ability of biomarkers, and it also contains inhibitory agents blocking signal visualization, especially for lateral flow assays. To overcome these problems, we created a new device comprised of three functional units: a collector, a unit with saliva quantity indicator, a saliva sample preparation unit, and a lateral flow-based detectors. These are protected by 5 patents and 11 pending patents and 2 trade secrets. On the left, you can see the collector. When enough saliva present in the sponge, a color change can be noticed in the indicator. Then the collector is inserted into the sample preparation unit, the saliva is squeezed out, treated, and reach the detector. Alternatively, the detector can be replaced by a tube, and the saliva can be processed but afterwards by a different detector. This new platform yield already several products regulatory approved in Europe, UK, and Australia, and was chosen as one of the best inventions by the Time magazine 2023. After selling this year more than 500,000 units, we looked at usability data as published in the web by several selling companies and compare it to our usability studies before launching the product. We could see nice agreements between usability studies and real-life results, as most women ranked the product 4 to 5 stars. We could compare it also to urine test and see that most women, and you can see it on the right-hand side, that most women preferred saliva assays rather than peeing on the regular test devices existing, emphasizing or encouraging us to continue developing our saliva-based platform. So, Omer, please.
Okay, thank you, Aaron. So Salignostics platform includes two product lines, Salignostics Om and Salignostics Medical. The Om line adapts existing rapid tests originally designed, as Aaron said, for blood, urine, stool, or swabs, for use with saliva, making them more suitable for at-home use. This is made possible by novel collection and processing technologies. Several tests shown on this slide are currently at various stages of development and represent just the tip of the iceberg of a much broader potential. The medical line targets complex diseases without established biomarkers. Here, we identify new biomarkers by cleaning saliva and exposing hidden proteins. Next slide, please, Aaron. As Aaron mentioned earlier, our first product is Salistick, a saliva-based pregnancy test for home use. This product enables the detection of pregnancy at very early stages with an overall accuracy exceeding 96%. In addition to being a novel product, it also serves as a proven platform for saliva collection and preparation. Its development has therefore laid the foundation for future assays. Next, please. Strep A is a common seasonal illness, especially in children, which requires antibiotics with a huge diagnostic market. Today, diagnosis relies on invasive throat swab, which are taken by professional healthcare providers unsuitable for home use. A saliva-based test would enable home testing, save time, improve compliance, and reduce sampling errors. In a preclinical study with the North Medical Center in Israel, we showed that Strep A can be detected in saliva using our unique processing method. Initial result showed that about 88% sensitivity and 96% specificity. The test is now being optimized ahead of clinical trials and regulatory approval. Next, please. In October, we ran a preclinical study in Ethiopia to detect HIV antibodies in saliva shortly after infection. Using a blood-based strip adapted for saliva with our processing method, we demonstrated promising feasibility. With 47 positive samples, we showed about 87% of sensitivity and 100% of specificity for HIV in saliva. Alongside our other tests, this again highlights the need for processing and filtering saliva to enable proteins detection. Next, please. Pancreatic cancer, part of Salignostics Medical Group, is a deadly disease often diagnosed late through invasive procedures. We're developing a non-invasive early detection test as a part of consortium supported by the Israel Innovation Authority. Saliva samples were depleted from alpha amylase, which is the most abundant protein in saliva, utilizing our amylase depletion column. Depleted samples were further analyzed by mass spectrometry, revealing more than 2,500 proteins. AI analysis identified 3 markers that accurately distinguish pancreatic cancer patients from healthy individuals, 2 previously known from pancreatic biopsies. While preliminary, these results demonstrate the importance of processing saliva for enhanced biomarkers detection. Next, please. We have established a medical-grade assembly facility equipped with dedicated semi-automated machinery, supporting production volumes of 1 million units per month. We are currently optimizing manufacturing processes to expand capacity and reduce costs in response to growing market demands. To conclude, next please. Processed saliva significantly enhance the analytical performance of rapid diagnostic tests. Initial clinical use in Strep A and HIV detection shows promising results with lateral flow assay. Novel saliva processing enables removal of interfering proteins and support biomarkers discovery. And finally, early sales demonstrate strong market acceptance, supporting broader application for home and professional saliva-based diagnostics. Please, next. Finally, I'm pleased to introduce our excellent team, our colleagues, Salignostics co-founders, Dr. Guy Krief, Dr. Raluca Cohen, and Dr. Yoav Neumann. Next. Thank you for listening.
Thank you for sharing that presentation. Really appreciate getting to hear about the work that your team is doing. Up next, we're going to transition over to hearing from Dr. Justin Rolando, who's joining us from the Wyss Institute of the Brigham and Williams Hospital, which is part of the Harvard Medical School. He's going to be sharing with us about advances in neonatal sepsis detection with ultrasensitive protein-based saliva diagnostics.
All right. Good morning, or good afternoon, or good evening, depending on where you're coming from. My name is Justin Rolando. I'm an instructor in pathology at Brigham and Women's Hospital and Harvard Medical School and a postdoctoral fellow at the Wyss Institute. I need to disclose that I receive royalties from Talis Biomedical for diagnostic tests I invented, and I perform consulting work for Chromologic, also in diagnostic testing unrelated to this presentation. This research in the presentation was supported by U01 from the National Institute of Child Health and Human Development. And I have this crazy idea. What if we stopped stabbing babies? What if we stopped stabbing babies? And you're probably thinking, but Justin, I don't stab babies. What are you talking about? That's ridiculous. And you're right, nobody wants to, but every day across the United States, the moment a baby is born, we do just that. Hospitals routinely collect blood for newborn screening, looking for basic genetic disorders and inborn errors of metabolism. And these are critical tests, but it comes at a cost. Oh, I'm sorry, I'm missing some slides here. But it comes at a cost. I don't know how this image makes you feel, but for me, it evokes a visceral, deeply unsettled feeling because you know what it takes to get this test done. And it can easily go wrong. You can bruise the heel, you can miss the draw, and meanwhile, a baby is crying and the parent is watching nearby. Instead of that parent getting to be focused on what should be a moment of joy, this new life, this new beginning, they're stuck worrying, is my child okay? Newborn infants, that's neonates, often have less than just 2 tablespoons of blood in their entire body. That's less than 30 milliliters. And as we've seen, they're fragile. These procedures can go wrong. On top of that, babies aren't just small, they're still developing. Their organs, their immune system, even their salivary glands are still developing. And that variability in gestational age makes everything more complicated. If you want to run a test for something like sepsis, a simple heel stick won't cut it. What you're going to need to do is go into an artery or a vein. And so we said this was a huge problem and assembled a team of researchers from the Brigham and Women's Hospital and the Wyss Institute, led by Professor David Walt there at the bottom right. We tried to answer the question, could we diagnose sepsis without stabbing a baby? So let's define what we're really talking about here. Sepsis itself is not the infection, it's the body's response to infection. So you can have bacteria in your blood, we would call that bacteremia, or fungi in your blood, fungemia, and not be septic. In fact, many of us had transient bacteremia this morning after brushing our teeth, and we didn't die, thankfully. And so because sepsis is the body's response, every baby has a risk for this, which is why it's so important to catch sepsis early. And the challenge here is that the symptoms of neonatal sepsis are incredibly nonspecific. Things like rapid breathing, jaundice, poor feeding, any one of those could be signs of sepsis, or it could just be the baby adjusting to life outside the womb. And I meant to say those. So rule out sepsis is the most common test in neonatal intensive care units. It's because of the uncertainty in the diagnosis itself. And so every year in the United States alone, we have about 1.75 million newborns that are tested for sepsis. It's nearly 2/3 of neonatal intensive care unit admissions. And most of these babies aren't infected, but we test because the stakes are just too high if we miss an infection. And so neonatal sepsis is the 3rd leading cause of newborn death worldwide, with over 200,000 deaths per year. Right now, if you're in the hospital, if we suspect sepsis in a newborn, we go straight to the blood culture. But this approach has some serious problems. It first requires a venous blood draw, and that's painful. Each test requires at least 1 ml of blood. And so for a baby that has only 30 mL total, every draw is a risk for causing anemia. If that anemia gets bad enough, they may need a transfusion, which also carries serious risks. In fact, about 30% of cases, these transfusions in the neonate can lead to a deadly complication called necrotizing enterocolitis, where the intestines rupture and the infant dies. And so after you collect that blood, then you wait. You wait, you wait, and you wait, and you wait, and that culture takes 48 to 72 hours to come back. That's time. That's time that you don't have because inside the baby, an infection could already be growing. And so as a result of this, as soon as the clinician suspects sepsis, they begin administering antibiotics. But the blood culture is deeply flawed and misses as many as 60% of actual infections. So when the result comes back negative, the physicians don't trust it. The physicians don't trust it, and as a result, antibiotics are often continued in about 20% of those putatively negative cases. And this also comes at a cost. Yes, antibiotics save lives, but we don't want to use antibiotics unless we absolutely need to. There are alternatives. Currently, there's PCR. We have this BioFire from Biomarker at our hospital. It's faster than blood culture and it's more sensitive. It relies on nucleic acids, obviously. And so the challenge here is that you can't distinguish between a living micronucleic acid and a dead micronucleic acid. And that leads to high false positive rates. It tests a limited number of common pathogens. It includes some antibiotic resistance genes, but it's missing others. So it's not universal. It's actually quite expensive as well, at $1,000 per test. And so here we're looking for the actual infectious organism. In contrast, another system we have at our hospital is this system from Roche that's looking for C-reactive protein. It's faster than blood culture. It's inexpensive relative to the other tests. But because C-reactive protein is just generalized inflammation, it's fairly non-specific. So to be precise, you have to do repeated testing, which means stabbing the infant multiple times. What I really dislike about both of these systems is that they require blood. That's something we're trying to avoid. So here's the thing that most of you who are parents already know. The one thing that babies are really good at is producing saliva. So we're leveraging something that babies naturally do well and something that their bodies haven't quite figured out how to regulate yet. Because in neonates, the salivary glands are underdeveloped, and that means that some of the proteins that would normally stay in the blood leak into the saliva. And this gives us a window into what's happening inside their bodies without ever needing to draw blood. And so we've recognized that saliva is a powerful biofluid, as many of you have today. The clinicians, the nurses, and phlebotomists don't mind collecting it. The parents don't mind their child being tested because it's safe, it's simple, and it's non-invasive. And that worked in our favor. We had nearly 100% consent rate in our clinical trial. But to make this test work at the clinical level, we needed to solve two key challenges. One, the analytical sensitivity, because we're working with tiny volumes and low concentrations. And two, that clinical specificity, because if we're going to make a decision to stop antibiotics, we have to be confident that we're right. And so this effort took a truly multi-institutional collaboration led by our Professor David Walt at Center alongside Professor Joe Marin, just to his left. We worked closely with 3 clinical partners, including Women and Infants Hospital in Rhode Island, the University of Florida, Gainesville, and Tufts Medical Center, each of these hospitals contributing patient samples and deep clinical insights. And so to make this test work, we needed more than just a good idea. We needed a technology that could see what no standard assay could. And so over the years, the last 10 years, the Walt Lab has developed an ultra-sensitive protein detection platform, which we call SIMOA, or single molecule array. It allows us to detect and quantify proteins in less than a single drop of saliva and in just a few hours. Let me walk you through what you're seeing here. Each tick on the x-axis is a different protein we found in saliva, and the y-axis is the concentration we observed. So you'll notice that there's a dashed line across the middle. That's labeled conventional ELISA. That's the typical limit of detection for existing standard technologies. The green dots represent the maximum concentration of a protein we observed, and the blue square is the lowest concentration we observed in saliva. The first protein, CRP, that's the one you may recognize from earlier. It's observable in some infant saliva. using conventional ELISA, but for most of these infants, it's undetectable. For the remaining biomarkers, you'll notice that their concentrations are far below what traditional tools can see, and this blue region represents previously invisible information only accessible using our technology. And so this isn't just a more convenient test, it's a fundamentally more sensitive one. And that opens the door to diagnostics that were never before possible. So when we took this new information, made visible by SIMO, we found something very exciting. We were able to see a biomarker fingerprint of sepsis begin to emerge. And so this is the largest neonatal salivary study ever done. In just the infants that were included here in the models, we have over 1,400 infants enrolled from 3 different hospitals. More than 2,300 samples were used and more than 55,000 total measurements were made. And again, that's emphasizing just the data that we include in our models, which excludes some 20,000 measurements from infants that had similarly manifesting diseases like pneumonia and necrotizing enterocolitis. What we're looking here at the graph is 12 different protein biomarkers that are found in saliva. And the left column, you'll see an uninfected group, and then on the right column, the infected. Along the y-axis is the protein concentration observed there in the saliva. And so now when we actually look at the data from these tests, we found that for every single protein we looked at, every biomarker, there was a statistically significant difference of the medians. And that's except for our negative control biomarker up there in the top right. And so if you were to take any individual infant, however, that's the single black dot there, what you might notice is that you wouldn't be able to place it into an infected or an uninfected group because there's so much overlap in those biomarker concentrations. And so when we combine these biomarkers with clinical metadata from over 400,000 infants, we had what we needed to train a machine learning algorithm because no single biomarker was sufficient to diagnose sepsis. And so we need to benchmark now our novel salivary diagnostic against the standard of care blood culture. And for those of you unfamiliar with these matrices, it's called the confusion matrix. I'm going to try to explain what we're looking at. It's how we compare the test's prediction to the actual ground truth. The rows represent what's actually true about the infant, whether or not they were infected, and the columns represent what the test predicted, so whether the test thought it was infected or not. So in the top left corner, we have a test that says correctly that the baby isn't infected, and they were not. And at the bottom right corner, that's a test that correctly identifies an infection. This is a good result. And the other 2 corners are then when something goes wrong. So how did our test perform? Let's just focus on the correct prediction. This is just one of many models that we've developed using saliva. In this example, we're using only data available at the time of the initial rule-out sepsis test. We also have time series models that track how the biomarker levels change with time during hospitalization. And this particular model correctly identifies 74% of infected infants, but more importantly, it correctly rules out 78% of the non-infected cases. And that's our primary goal. It's to quickly and confidently identify the infants who are not infected and who do not need antibiotics. How does this stack up against blood culture? Again, let's just focus on the correctly identified cases. And so across the board, we see that our models outperform that standard of care blood culture. We see a 14% improvement in identifying uninfected infants, that's the top left, and a whopping 34% improvement in identifying infected infants. And so as a result, if you extrapolate that across just the United States, we estimate that our test could rule out and prevent unnecessary antibiotic administration in up to 1 million infants per year. And so our vision of the future is this, that sepsis, one of the most feared and challenging neonatal diagnoses, can be detected non-invasively using saliva. We believe that our approach can reduce unnecessary antibiotic use, minimize infant pain, and ease the anxiety that weighs so heavily on parents in those early critical days. And I'll end where we started, because if we can show something as complex as diagnosing sepsis can be done with saliva, then maybe we can move other diagnostics into this fluid too. And maybe, just maybe, that means we can stop stabbing babies. I'd like to make a note that Kathleen Bellin Pizarro is presenting a poster just after this session on the ultrasensitive detection of 6 sepsis-associated proteins in neonatal saliva. That's part of this biomarker discovery manuscript, which you can scan the QR code to get a direct link to. That's proteins that were not included in these models and in this study that I'm presenting today. If you'd like to learn more about my research, there's a link to my website there. Thank you for your attention.
Thank you so much, Dr. Rolando. Appreciate you sharing about that, your topic, and also highlighting the upcoming poster session and your student that's going to be involved. Next, we're going to be transitioning over to Dr. Aaron Weinberg, who's joining us from Case Western Reserve University. He's going to be sharing with us today about the BDI oral cancer biomarker from ELISA to rapid test.
I'd like to thank the organizers for inviting me. I had a wonderful time last year, and I think this is an outstanding organization. I'd like to continue supporting it if possible. So I'm going to transition from the liquid in our mouths to the cells in our mouths. And I'm going to tell you a little bit about work that we've been doing in an attempt to create a point of care rapid test for the detection or early detection of oral cancer. And if anyone is interested in communicating with me at any point in time, this is my email address. So as an introduction, head and neck squamous cell carcinoma, of which oral cancer is about 90% of that entire umbrella name for multiple different types of cancers of the head and neck. But oral cancer makes up 90% of it. It's 6th or 7th most common cancer known. There are about 640,000 new cases each year with 200,000 deaths annually in the world. And in the US, it's about 11,000 deaths. It's caused primarily by smoking, alcohol use. Now more frequently, you're seeing in younger people, HPV-related oropharyngeal squamous cell carcinoma. And something that you uniquely see in the subcontinent, in low middle income countries, in the ASEAN countries, Taiwan, southern China, the use of something called betel quid. And that comes from the areca tree. The betel nut is something that's been used for centuries in these people. You'll see a characteristic kind of red-orange dye in their mouths after usage of it, but it's supposed to generate a feeling of euphoria and awareness. It's like our Red Bull times 10. But over time, it can create what's referred to as submucous fibrosis, which can lead to squamous cell carcinoma in the oral cavity. The gold standard for oral cancer diagnosis is a scalpel biopsy followed by pathology review. Its limitations are that it's costly. Depending on what medical center is performing it, it can go from $1,500 to over $3,000. It's invasive, obviously, can be associated with certain patient morbidity. And in LMIC countries, the pathology reviews are often times imprecise and misreported. It's clearly not feasible if one wants to follow with repeat assessments of a given suspicious lesion, monitoring it over time. And in the Western countries, clinicians tend to over-biopsy. Mea culpa, I used to do the same thing, so I know what this is about. They want to make certain that they cover their bases, so to speak. So the clinicians tend to over-biopsy. And greater than 90% result in non-OSCC outcomes, thankfully. So the unmet need here is for a rapid, accurate, objective non-invasive, low-cost detection of oral cancer that could assist the clinician in knowing when a biopsy is warranted. So as an introduction, I'm going to tell you a little bit about work that my lab has been doing over the last several years. When I was in practice, these sorts of lesions that one would see in the oral cavity, routinely self-inflicted lesions, like biting your cheek or biting your tongue, or in response to anesthesia performed by a dentist, you'd get these lesions that would compromise the mucosal barrier. And I was always intrigued by how it is that we don't get bacteremias. We don't get bacteria, fungi, or viruses, and we know that there are multiple different species of bacteria in the oral cavity that attempt to get into these lesions and raise havoc. So my lab started looking into these particular kinds of lesions, and we discovered small peptides, which later were referred to as beta defensins. And these are peptides that are produced by adjacent epithelial cells in response to the challenge. And these we discovered were antibacterial, antiviral, and in certain cases, antifungal, and in certain cases with encapsulated viruses, they were also antiviral. And so that started a whole area in my investigation of what these molecules actually do. So they're evolutionarily ancient. They were first discovered by entomologists in the fruit fly. They're very conserved. They're produced exclusively by epithelial cells. They're beta-sheeted, very positively charged, highly cationic, and they interact with anionic sites on the bacterial surface, such as lipopolysaccharide, lipoteichoic acid, the phospholipids of the bacterial membranes. And once they attach, they're also amphipathic, which means they have a hydrophobic and a hydrophilic domain that embeds them into the bacterial membrane, and then eventually pores form and the organism implodes. We discovered over the years these different permutations of the beta defensins: human beta defensin 1, which is constitutive, 2, which is inducible through a transcription factor referred to as nuclear factor kappa B, and HBD3, which is inducible through EGFR, epidermal growth factor receptor, and I'll come back to that because this is pivotal in my presentation. And then there's HBD4, which is also inducible. But the key thing to remember here is that there's a big difference between these two. HBD2 is induced through NF-kappa B in response to and supporting inflammation. HBD3, on the other hand, is inducible for wound healing under normal conditions, and again, is induced through the activation of EGFR. Now, the reason I mention this is because EGFR is central to oral cancer proliferation, oral cancer production and proliferation in an uncontrolled manner. So just keep that in mind. And here, what I want to point out visually is what we're seeing when it comes to human beta defensin 3 and its correlation or its association with early stage cancer. Here you're seeing fluorescent immunohistochemistry of the floor of the mouth in a normal individual. You're seeing a basal layer, a spinous layer, a granular layer, and a stratum corneum, which is the keratinized layer. And what happens is cells that emerge, and they're starting as stem cells that emerge at the basal keratinocyte layer, the basal layer, they start to ascend. But in this region, they are highly proliferative and non-differentiated. As they ascend into the spinous and granular layers, they start to differentiate, stop proliferating, and then go through a cycle of death. And this thing repeats itself every 7 to 14 days, depending on what mucosa we're talking about. But what I want to bring to your attention is that fluorescent antibodies specific for HBD3 will routinely label the region of the highly proliferating part, the basal keratinocyte region of that mucosa. And it'll stop producing HBD3 and start producing HBD2 as the cells start to differentiate. So that's under normal situations. Now, here what you're seeing is something that we found really fascinating, is that in the context of carcinoma in situ, which is stage 0 of oral cancer, the earliest sign of oral cancer, the HBD2 is, in this case, almost completely gone, and there's only HBD3 expressed in this entire lesion. We also know through PCNA labeling that these areas, this representing this area here, and this area representing the carcinoma in situ, is highly proliferating. Okay, so when you take a green fluorescent tagged antibody with a red fluorescent tagged antibody, you're going to get orange. So all this area here is very, very colored in orange. And then when you look over here at the nuclei in the region of the carcinoma in situ, there is an agent called beta-catenin, which is from the Wyss system, and it translocates into the nucleus. That is a signature signal or sign for cancer in this lesion. So you can see how the nuclei differ here than they do under normal in the normal area of the tissue. So this and this and this are indicating to us that this is carcinogen, that this is cancer, and that there's overexpression of HBD3. Well, and this is just another example of what we found. Stage 2 squamous cell carcinoma of the tongue. Under normal tongue tissue, you'll see a lot of HBD2. And in this case, with SCC stage 2, there's a lot of green represented for HBD3. Another thing that we found with regard to HPV-16, which is the human papillomavirus strain that is very much associated with oropharyngeal cancer, you'll see here also a lot of HBD3 expressed in the cells or in the cytoplasm of the cells, as you can see here and as you can see here. So both squamous cell carcinoma, both carcinoma in situ and HPV related oropharyngeal cancer have this common phenotype. And that's what I'm saying up here. HBD3 overexpression is a common phenotype in oral carcinoma in situ, oral squamous cell carcinoma, and HPV-related OPC. I don't have time to go over all of this stuff that we discovered over the years through the publications, but aside from that, I want to bring to your attention the fact that HBD3 we discovered is not only an antibacterial agent, but it's also a chemokine, and it recruits tumor-associated macrophages to the lesion site. That is a bad thing. And I'll talk about that later nearing the end of the presentation. But just remember that HBD3 also acts as a chemokine exclusively for these macrophages. We also discovered through HPV-E6, the oncogene that suppresses P53, the very important suppressor of cancer, so that when E6 gets activated, it suppresses P53. Turns out that that modulates HBD3 expression to go up. So those are three things that I think are imperative, very important in the context of understanding the role of HBD3 as it relates to oral cancer. So last year, we published this paper that appeared in Cell Reports Medicine about a beta-defensin index, a functional biomarker for oral cancer detection. And what we did here is we did multiple studies, proof of principle studies, a discovery blinded study, and a 3-site validation study, where we tested the concept that if we were to take cytobrush samples from the lesion site and a paired cytobrush sample from the contralateral normal site and conduct ELISA assays on these samples and eventually conduct through this formula referred to as BDI, or beta defense index, where the OD of HBD3 over the OD of HBD2 of the lesion site over the OD of HBD3 over the HBD2 in normal sites, so it's a ratio, the ratio gave us an index. It turns out that the discovery study through ROC curves demonstrated that the BDI index, anything above 1.25, was demonstrating cancer. Anything below that was non-cancer. And through all these studies, we identified almost 100% sensitivity. The specificity was 83%, so that the 17% false positive rate, it turns out, was due to, and this is very important, the highlight of the BDI being able to identify precancerous lesions. The precancerous lesions, by definition, are considered non-cancerous. But for the clinician, having a tool that enables him or her to be able to identify precancerous lesions, not just cancerous lesions, I think is a major plus. So I'm putting that in as a caveat when talking about the specificity. So what we want to do now is pivot away from the ELISA into a point of care device for early detection of oral cancer. And the biomarker, again, is based on the ratio of two proteins, HBD3 and 2, taken from the lesion and contralateral normal site in the same patient. We envision that this can be performed in any dental clinic as part of oral health checkups for early detection. And additionally, we envision that the results could be obtained in 15 minutes using a handheld device. The salient features of the assay is minimally invasive. I think that's very important. It relies on 2 analytes, not 1. Very importantly, the subject is their own control. We don't need the assay system to require absolute concentration, eliminating the necessity for the inclusion of a standard analyte of known concentrations in every assay. It provides an objective quantitative measure, and both the normal and lesion sites can be analyzed simultaneously, minimizing interassay variability. So those of you in the lateral flow arena see this as very simple. For me, it was an eye-opener because I'm not a lateral flow aficionado, but my colleagues are. And what we're trying to do here is design and prototype a lateral flow fluorescent immunoassay test where the sample comes in here and goes through this process of being identified through conjugated europium antibodies to HBD3, to HBD2, and control antibodies, so that we can then take that, use a fluorometer, and be able to identify the levels of expression of 2 and 3, both in the lesion site and in the contralateral normal site. So that our intention, as we study this, is to take samples from individuals. We have a lysis buffer that will lyse the cells and simultaneously protect them against multitudes of proteases, and then run them in the rapid test, as you see here, and eventually read them through a mobile readout. And this is in conjunction and collaboration with Hemix Health, I'll talk about in a moment. They have a Gazelle system, which they have used for fluorescent immunoassays of lateral flow for hematological purposes, such as sickle cell anemia. And we intend to test this in their system. So what's in front of us? What are we planning to do? And what are we doing right now? We were fortunate to receive portent funding, which was only recently released to us, with the intention of conducting analytical testing and optimization of the rapid test, and then eventually testing it in healthy and lesional cytobreath samples. We're working with a gentleman by the name of Kevin Jones from XHG out of Chicago. He is an expert in lateral flow technology. We're fortunate to have him on campus. One of the labs is associated with us here on campus at Case Western. We're also submitting an R01 academic industrial partnership grant where the industrial partners Hemix Health in August, and our collaborators, our clinical collaborators, are at Case Western University Hospital's Rod Razai, who's been working with me for a number of years and was a co-author on the Cell Reports Medicine paper. And we're also including Alessandro Villa from the Miami Cancer Institute, who is an oral medicine specialist. Rod Razai is an ENT surgeon. And finally, we're also working with India Institute of Medical Sciences in Bhopal, India. And those who know, Bhopal is an epicenter for oral cancer in India. So it makes a lot of sense to be working with these experts there. Other potential applications. So you may recall that I talked about the ability of human beta-defensin 3 to act as a selective chemokine for macrophages. Now, in the context of oral cancer, myeloid-derived suppressor cells is a bad sign. When they come into the lesion, they will suppress T cells that are specific in trying to kill the tumor. These bad guys are a bad sign, and prognostically, they're indicating that the lesion is going the wrong way, so to speak. So what we are doing also now is when we take the cytobrush samples, we're quantifying MDSCs using the same principle, and we're looking and comparing the correlation between BDI scores and the MDSCs. We think we might be able to provide the clinician additional information about the status of that lesion. We're also looking into tonsillar or pharyngeal lesions because our preliminary data indicates that in sedated patients, doing this procedure has merit, because we've been able in a proof of principle study that appeared in the Cell Reports paper that we can pick up oropharyngeal cancer this way as well. And finally, BDI could be used in detecting other squamous cell carcinomas because the common denominator is overexpression of HPD3 in cervical, anal, and esophageal cancer. The team that we've put together, it includes Umut Gurkin from the School of Engineering. He has a lot of experience in hematological related diseases using lateral flow. He's been working with Peter Galen from Hemix Health. And what's interesting about Hemix Health is that it focuses its main attention in low middle income countries, in Africa and in India, sickle cell anemia being one prominent disease that they're focusing on identifying. So we think this is a really good match for what we want to do and what Hemix is all about. And finally, Santosh Ghosh, who's been working with me for almost 20 years, is the person who perfected the ELISA assay that started this whole process. And obviously, NIH and CWRU funding over the years that permitted us to get to where we are today. And with that, I thank you very much.
Thank you, Dr. Weinberg. Appreciate you sharing about what your team has been working on and where you all are thinking about heading in the future. With that, we'll transition over into the Q&A portion of today's event. And we have had a couple of questions that came through, and so I'll just go through them in the order in which they were received. The first one is actually over to Dr. Rolando, and that is looking at all of those biomarkers that your team was able to identify. What else are you looking for or able to detect from that wealth of data, all that information that you have to now mine? What are you looking forward to?
Certainly. So I understand the question to be, we have a plethora of additional data in addition to the proteins. There's electronic medical record data. There is an alphabet soup of diseases associated with neonatal sepsis, necrotizing. So there was NEC, there was pneumonia, there was rupture of prematurity. There's a lot of things that I think we can start to look for. So our preliminary work is to try to unravel if we can identify sepsis itself. And then from there, we're going to look at correlations with other disease states.
Thank you. Appreciate that. And a follow-up question for you that came through related to this is, what are the biggest challenges that you foresee in translating this non-invasive saliva-based sepsis test for research into routine clinical use in NICUs?
Yeah, so I think one of the great things that we saw from the Salignostics team, from the Case Western team, was that they have portable devices that they're able to bring into the clinic directly. What we lack is a device that we can translate directly into the clinic. So the NICU is especially small. The physicians don't want another printer-sized box on their tables. They need small things, shoebox maybe, or smaller, and it needs to be easily and operated by the nurses. So we're looking towards developing point-of-care devices, point-of-care instrumentation. And then additionally, one of the things that we really rely on is that we have an ultra-sensitive detection scheme. It's using the same techniques as ELISA, but we improve that by doing single molecule counting. Scaling that down into the clinic needs to be done. I think we can translate to other similar platforms like the high throughput Roche instrumentation that we have in hospitals. But again, it's a commercialization challenge at that point.
Absolutely. One of the questions that came through that relates to that perhaps we can have Dr. Weinberg answer and then open it up to other panelists is the cost differential. Each of you had mentioned just how expensive current costs are or how challenging they can be, particularly Dr. Rolando and Dr. Weinberg, you mentioned thousands of dollars for the current standard of care. And so there was curiosity about just how much of a cost savings potential is there with these technological advances that you're proposing?
Do you want me to answer first? Okay, we've done a deep dive into this back when we were trying to get some funding, and I had to put on a suit and stand in front of potential people interested in this whole thing through Coulter. We have a Coulter Foundation, and so I learned what unmet need was, and I learned about the importance of cost savings and all of that sort of stuff. So we did a deep dive into this, and I feel that what the cost would be for the rapid test for oral cancer would be somewhere in the vicinity of $50, $60. And clearly an LMIC, is that what it's called? LMIC. So in LMIC countries, it would be a fraction of that, especially in India. And we're headed in July through Portent to India. Maybe you know this. I mean, you guys are into Portent and all that sort of, acronym thing. And so we're very fortunate to get the funding through Portent. But part of it is a trip to India, to Bangalore, to meet with all kinds of people, from health-related individuals, health department-related individuals, academics and all. We'll get a much better understanding of what the cost can bear in those countries, in countries like India. But I foresee a fraction of that. Could be $10, could be $5. I mean, to manufacture these things is a matter of pennies. So, right?
Yeah.
I mean, you want to have a few shekels in your pocket, right? But at the end of the day, but honestly, I think in low middle income countries, which this is primarily geared for, sure, we want to see clinicians like myself, primary care clinicians, use it in the West for purposes of identifying who actually needs a biopsy, right? Because that can save a lot of money. Just that alone. But in low middle income countries, I'm hesitant to say, okay, everyone there can get a biopsy and pathology review. I don't know if that's possible. I'll learn more when I go there. But I think less than 50 in the West, a fraction of that in the East.
Thank you. Thank you.
I see my colleague Aaron Palmon moving his head like this, which is very encouraging. He knows more about this than I do. So,
Dr. Palmon, that's a great segue over to you. Is there anything else you'd like to add about this for your technology or where you see these opportunities for salivary diagnostics?
Yeah. I mean, these were very interesting presentations. And at the end, you need to have a device going to the market. And from our experience, this is a true challenge, okay? And of course, the market asks for low-cost production, and low-cost production, especially for such countries like India, China, and so on, the prices in the West cannot be compared to the prices there. And even in the Western countries, they don't want to pay a lot of money for such devices, especially if they are going to not to point of care markets, or if it is not covered by the insurance companies. So in these cases, these are price sensitive, and it depends on the alternatives that exist. So if the alternatives cost a lot, like a biopsy, okay, so you can increase the price of your device as you can. Okay, and of course, if you develop something for 10 or 20 years, you deserve to get something in return. And that's it.
One last thing I want to say is we also did a deep dive into insurance codes, and there are 3 existing codes in the U.S. for cytobrushing in the oral cavity. So that would be covered.
Yeah.
Interesting.
And the translation into the market, it's not so simple. It should be robust, without mistakes in the procedure itself. And it's a big challenge.
Absolutely. Dr. Rolando, your thoughts on cost or the differential between what you're proposing and what's on the market, or just getting things out into the market itself?
I think as both of these, my colleagues have stated, there's a large difference in cost to manufacturing versus what's billed to insurance. And their entire team's dedicated to determining what that goes into the cost to insurance. And I think there have been a lot of interesting innovations, especially in subsidizing costs to bring them to low and middle income countries so that we don't have to see these extremely high price points like we have in the West. And I'm not really sure what determines outside of market economics, the absolute cost billed to insurance, and how we make these things more available and equitable and easily disseminated.
Absolutely. Hopefully, as these technologies become more well-known and more into awareness and just the mainstream, it'll move things in a direction where it will become the expected, that you'll be able to have something that's non-invasive and less expensive than what a lot of folks are used to or what have been used in previous generations. Absolutely. There's a question that just came through that's actually related to this and thinking about getting things to market, and that's related to regulatory goals. And so if each of you wouldn't mind sharing what regulatory goals you have for your products that are identified for distribution in the US, if that's your primary location, or elsewhere, recognizing that you all might be looking at other markets as well.
Okay. We have experience with several countries, not in the US because of the cost. Okay. So we got the CE from the EU, from UK, and TGA from Australia. So we are already selling the products. Okay, for the US market, we prefer to have a big company collaborator and only then submit a full FDA. Okay, we need to know that the FDA today requires 95% sensitivity, though older FDA-approved kits were with lower sensitivity, but nowadays they ask for 95. We made a pre-sub for the Strep A, okay? And the Strep A is very sensitive to the method how you collect the sample. So most regular swabs, okay, throat swabs, if you look on the published scientific papers, are much less than 95% because they are very technical sensitive. But nowadays, if you want to submit a new submission for approval, they ask 95%. So when I looked on all these nice approaches presented, I thought to myself, okay, let's go to FDA. Okay, so we need to understand that the rules are changing and our merits should be elevated to the rules. Otherwise, there will be more problems in the approval process. But currently, we went to other markets because of the cost. The FDA requires that clinical trials will be mostly in the US, and the price of clinical trials within the US are much more than other countries. So you can't use almost any data you have previously gathered from other clinical trials to the FDA. So these are complications, and therefore you need big companies alongside to your developments, unless you are a big company by your own.
Absolutely, absolutely. That's something that our own team is experiencing as we're making our headway through going through de novo applications and getting our technology through those systems, those requirements for not only the sensitivity, but also all of the clinical samples. That's our rate limiting step perpetually is those samples. Dr. Rolando, what about your team in terms of regulatory goals or progress made to navigate the FDA system or otherwise?
We are still actively soliciting partnerships from, let's say, larger commercial vendors who would be able to support our manufacturing and regulatory goals. We're still a research lab trying to discover new biomarkers of disease and ones that will be easily combined. We have a great support network system across the hospitals for obtaining samples. So anyone who wants to partner with us there, that's a great opportunity. But we don't have that expertise in regulatory and manufacturing.
Certainly, certainly. As an organization that spun up from a university, we certainly appreciate the connections that builds in. And I'm going to make a note to check back in with you about samples because we always love those kinds of partnerships. You never know. You never know. Dr. Weinberg, what about your team in terms of regulatory goals or hurdles that you think would be helpful for this group to hear about? Oh, you're on mute right now.
My apologies.
There you go.
I'm in the Rolando camp because Palmon is way ahead of us. He's been at this, I know, for years, and hats off to him, and I may even turn to him for some advice. But, you know, we've got tech transfer here, just like Rolando has at his institution, and they're guiding us in regards to this. We also have this collaboration with Hemix Health. They've been around for a while. They know the ins and outs of FDA regulations. I will tell you, we have CE mark. I know that covers that in Europe. And I think we've got it also in India and some other countries in Southeast Asia. And I know that we're going to have to file something related to predicate devices. You know, again, I'm learning as we speak. So is this unique? Is it not unique? That's going to dictate to a great degree how we're going to go forward with this, right? So stay tuned as we develop this. I will tell you that we have a grant in to the NIDCR for a clinical trial, a big clinical trial in the US, which is to a great degree patterned after what FDA wants. So if we can get this clinical trial in, with the data that FDA wants before FDA tells us this, we're going to look really good, right? So we're trying to get funded to do a multi-site US study on the BDI based on the ELISA, okay, before we go anywhere with the point of care, based on the ELISA, so that we can confirm it. And the assay is going to be conducted by a CLIA lab, independent of us. And I don't need to tell you how important that is. I mean, I would have preferred that each site would do its own assessment, because at the end of the day, that's what's going to happen. But it was prohibitively expensive in the grant to do that. So we had to do one. I think we did one better in that all the samples are sent to a CLIA lab. At the end of the day, maybe that's what's going to happen if it's ELISA-based. So
Absolutely.
that's where we're at right now.
Absolutely. I appreciate you each sharing about where you're at in terms of the regulatory challenges and where you think things are going. And it's really striking to me something that is consistent throughout each of your responses related to the partnership and how you're looking to establish these relationships with businesses and researchers and navigate these regulatory challenges that many of us on this call are experiencing in our own teams and with our own research that we're trying to get out there and connect people with. And that's something that we've certainly come to value from these conferences, particularly our conference that we're hosting, is connecting individuals who are working in similar veins and maybe we can learn from or partner with, or at least have someone to grab a coffee and be like, I'm with you on this too. You're in good company and in good shape. I want to be mindful of the time that we have, and we're just about at the end of our allotted time for this second session, but really, really appreciate the presentations that were offered. Thank you, Dr. Palmon, Dr. Rolando, and Dr. Weinberg for sharing your insights and expertise.